Okay, so we're going to take a few minutes to talk about energy metabolism in some of our organs. So this is the last slide of the integration of metabolism lecture. This is slide number 52. It's a table that we're just going to fill in, and I'm just going to talk through about the organs that are undergoing these processes. Vertebrate, looks like an A. Okay, so let's start with the brain. So the brain, energy reservoir, meaning does this organ store its own energy? And the brain does not have any stored energy. The preferred substrate for brain is glucose when glucose is available. However, it will use ketone bodies, which we haven't talked about yet, but during starvation, we can create what are called ketone bodies. Ketone bodies can cross the blood-brain barrier and actually be used as an energy source for our brain. Ketosis we'll talk about a little bit more later when we talk about fatty acid oxidation and protein degradation. So we'll see more about this later. Exported energy sources are also none for the brain. The brain is not exporting any energy. Let's see what else to say about the brain. So the brain definitely uses the most oxygen out of any, well, probably per body mass. The brain is only about 2% of our body mass, and it uses about 20% of the oxygen that's consumed. It uses a lot of the glucose to make ATP, and it needs that ATP to power the potassium-sodium ATPase action for transmission of nerve impulses. So the brain requires a lot of oxygen to, and it uses a lot of the ATP. And again, we'll talk more about ketone bodies when we talk about proteins later. So let's talk about skeletal muscle. So there's two situations here. There's resting and then prolonged exercise. So resting skeletal muscle, this is where our glycogen is stored. Glycogen is stored in our skeletal muscle. It's also stored in our heart. I'm gonna go ahead, oops, I forgot heart on here. I'm gonna put heart. It's also stored in our heart and used for energy, and in our liver. So remember what glycogen is. Glycogen is stored glucose. So resting skeletal muscle will use glycogen for energy. Its preferred substrate is fatty acids, which again we'll talk about next. And it does not have any stored energy. Let's kind of compare and contrast resting versus prolonged. I'm going to draw some lines in here. Again, this is all in your lecture, and it will look a lot better on that slide. Whoops, heart, adipose, liver, kidneys. Okay, so let's look at the two types of skeletal muscle, resting versus prolonged exercise. So prolonged exercise does not have any glycogen left. Its preferred substrate is glucose, and its energy source exported is lactate. Lactate is just the conjugate of lactic acid. So you'll hear people say when your muscles are sore that is stored up lactic acid in your muscles. And that is a little bit true. Lactic acid is it will make your muscles burn, and that's the actual lactic acid that's being produced. So if you're sprinting up a hill and your legs are burning, that's the lactic acid that you're feeling. But lactic acid and lactate are cleared from your muscles in about 30 minutes. If your muscles are sore the next day, that is not from lactic acid. That's something called delayed onset muscle soreness. That's your muscle fibers tearing and rebuilding. So lactic acid is responsible for that burning feeling when you're exercising, but it's not responsible for sore muscles later. And that's something that you commonly hear misinformation about. Let's see, what else? So your muscle is going to be undergoing glycolysis to produce pyruvate. One of the fates of pyruvate that we said was lactate. So then that lactate then can go to the liver and be converted back into glucose through gluconeogenesis, and then it's cycled around in the Cori cycle, which we're going to talk a lot more about when we talk about muscle, when we talk about proteins. So this is how muscles use what their energy sources are and what they use for fuel. The heart is a completely aerobic organ. It also uses glycogen for a energy reservoir. It also can use fatty acids or glucose or ketone bodies. Whoops. Let's see here. Fatty acids here. And it doesn't have any exported fuels. Adipose tissue, which is fat cells, its energy reservoir is what's called triacylglycerol. This is a molecule that we store in our adipose tissue, and this is fat. We store it in adipose tissue, which is made up of adipocytes, which are fat cells. The preferred substrates are fatty acids. And its exported energy sources would be fatty acids and glycerol. So we'll talk more about these molecules when we get to fatty acid synthesis and degradation. The liver can also uses glycogen and triacylglycerols. Its preferred substrate, it can use anything. The liver, you'll see, is often called the metabolic hub of our bodies. It does all the metabolic processes. Some of these other ones can only do one or a few. Pretty much every metabolic process that happens in our body happens in our liver. It can use amino acids or glucose or fatty acids. And it can export almost everything too. Fatty acids, glucose. Remember, it can export glucose because the liver and the kidneys are the source of gluconeogenesis, where you are making glucose from a non-glucose precursor, and ketone bodies, which we'll talk about. That can also be fuel for our brain in times of starvation. Let's see what else to say about the liver. A lot of regulation happens in the liver. Since all of these metabolic processes are happening in the liver, different hormones and second messengers are regulating which processes are happening and which are not happening. And that is all dependent on intracellular concentrations of different substrates. Let's see what else to talk about here. The last one, I'm not sure if I talk about the specifics of kidneys, but kidneys, the major purpose of the kidneys are to filter our blood. To remove urea from our blood. So we're going to see when we talk about denitrification. We're removing that amino group from amino acids. We need to get rid of that urea because urea is toxic to us. So the major function of our kidneys is to produce urine, to excrete. So we can remove that because it's toxic if it's not. So during starvation, it also is the site of gluconeogenesis. So it can export glucose. Kidneys also use a lot of our oxygen. They only are about 0.5% of our body mass, but they consume about 10% of our oxygen. Energy reservoir, there is a little bit of glycogen in our liver. Mostly it's in our kidneys. Its preferred substrate, it can use all of these too, like your liver: amino acids, glucose, fatty acids. And I know at this point we've really only talked about sugar metabolism, but we're going to talk about metabolism of fatty acids and proteins and how they are all regulated, and we'll refer back to this when we get there. And then we'll also talk about nucleotide anabolic processes, how we form nucleotides, and then how we use those to build nucleic acids like RNA and DNA.